So I'm Leonid Kruglyak. I introduced myself yesterday. I'm the chair of the Human Genetics Department here at UCLA. I'm the replacement speaker for Jonathan Flint, who is traveling. As a liaison, I will tell you I'm a really avid bike rider, so I don't want to keep you guys from your outing, and I'll try to get you out of here expeditiously. So my lab works on the genetic basis of phenotypic variation. So basically, the question is, if I sequence any two of your genomes, which is now a pretty routine exercise, and line them up, I will find a few million differences between them. And if I measure everything there is to measure about you guys, look up your medical records, give you a personality profile, etc., there'll be a long list of differences that will show up there. Many of them, if not all of them, have a strong genetic heritable component to them. So how do we connect those two things up? The differences at the level of the genome sequences and the differences at the level of the organism. And my lab is interested in these questions from a pretty fundamental basis. So we do a lot of our work in C. elegans, which I won't talk about today, and in yeast, which is our other favorite model organism. And we really focus on methods development employing combinations of new technologies and computation. So what I thought would be fun for this group is to tell you about a couple of methods we've developed over the last year or so, involving CRISPR-Cas9 technology. And the work that I'm going to talk about has largely been carried out by Meru Sadhu, who is a postdoc in the lab, and Josh Bloom, who is a staff scientist in the lab. Mostly on the experimental side and mostly on the computational side, although everything is through partnership from start to finish. Just to kind of set the stage for this, how do we usually connect variation at the DNA sequence level to variation in traits we're interested in? So for example, I'll illustrate this in yeast. You have two different strains, and one is high in some trait, some metabolite production, another is low. They have differences in their genomes, illustrated here in purple and orange. You cross them, generate a whole bunch of progeny after meiosis, which reshuffles these genomes. And then you score them all for the trait you're interested in, and look for regions of the genome where the genotype, in this case purple, is correlated with the higher values of the trait than the other genotype, in this case orange, right? So I've illustrated it here, and then you know that this interval for which this is true, and it's some interval, it's not a point in the genome, contains a variant or variants in some genes that influence your trait of interest. And I've illustrated this here in this context of yeast genetics, but it's exactly the same for mouse crosses or human pedigrees or even human GWAS, with the only differences being kind of the exact nature of this cross and over how many generations does recombination break up these segments, right? So this is really kind of genetics 101. And the role of meiosis and recombination here is pretty much taken completely for granted. But if you think about it, it has a couple of important disadvantages. One is that this region is pretty much entirely determined by two things. One is the size of your study, which you have some control over, but is usually limited by either availability of the subjects or money. And the recombination rate, over which you have no or little experimental control. So the region can be quite large, and getting from the interval to the actual causal variant is a huge challenge, as illustrated in humans by the fact that we have literally thousands of GWAS associations with different diseases and other traits. And you can list on the fingers of one hand convincing causal variants that have come out of those studies. I think that's not an exaggeration. And also, of course, the fact that you have to do this crossing and put these strains through meiosis, there are quite a number of interesting situations involving, for example, hybrids between closely related species, where You can do this first step of mating and producing progeny, but those progeny are typically infertile. So mules are kind of a classic example of what happens when you cross a horse and a donkey. And then you can't do genetics, even though there are lots of interesting trait differences in those types of scenarios. You can also think about, for example, if these are human cells from two different individuals and they have some interesting trait differences and genome differences, you can't mate them. So what we worked on is an alternative approach that then relies on sort of recombination-like events that take place during mitosis, so normal cell division, as opposed to meiosis, the special like reductive cell division during sex. So if you have a diploid cell and you make a double-stranded break on one of the chromosomes, one of the things the cell can do to repair it is to basically repair it off the other homologous copy of that chromosome. And once you kind of sort out what happens after mitosis, you can end up with these events that are called loss of heterozygosity, where you had a heterozygous diploid cell here, and then here you have a cell that for a chunk of that chromosome from some point out toward the telomere becomes homozygous for one of the two genotypes, starting parental genomes. So this is something that has been discussed in the literature for quite a while, and it's a known process that takes place naturally, but it takes place at a pretty low rate, and it's hard to identify these events or use them in any sort of targeted manner. So like many things in biology, this changed with the introduction of CRISPR-Cas9, right? So just to remind you guys, Cas9 is a nuclease, and the thing that really distinguishes it from other nucleases is that it's an RNA-guided nuclease. So you can supply an guide RNA sequence with a targeting sequence, and Cas9 will cut in a location in the genome that matches this targeting sequence. So it's sort of very precise molecular scissors. So as a result, you can do this process, but instead of waiting for these double-stranded breaks to just happen in cells, you can target them at high efficiency and with exactly where you want them using a guide RNA, and then you can target them in a different spot using a different guide RNA. And as a result, just like in the previous slide, you can have a panel of these mitotic, we'll call them progeny here, where the genotypes are heterozygous from the centromere towards some point on the chromosome, and then homozygous for one or the other parent past that point. And I can walk that point along the chromosome by design and place them as densely as I would like. And then once again, I can measure the phenotype and now also correlate the point at which having this breakpoint and going from heterozygous to homozygous changes the value, the typical value of the phenotype. So to pilot this whole idea, we used one arm of one of the 16 yeast chromosomes, the left arm of chromosome 7. And in this case, we used two strains that we've used in a ton of our experiments. They differ by about one SNP every 250 or so base pairs, so a little higher diversity than two typical human genomes. So one of the key things here is that these target sites are allele specific. So you only cut one of the two chromosomes, and that's what guarantees this exchange repair from the other one. So we made about 100 of these target sites that are specific to one or the other strain, and generated about 400 of these colonies. We can genotype them just by low coverage sequencing of these strains. And you can see for one of these strains here pretty clearly, you're getting reads that match the two different parental genomes at the sites where they differ from most of this chromosome. But then here you have a region where it goes homozygous in the direction of one of the parents. Here's a different place where we targeted the break closer to the centromere. So you have a larger region that's made homozygous. And in this case, we targeted it to be homozygous for the other parent genome. I'm going to skip this. So we have as our model phenotype sensitivity to growth in the presence of manganese for these cells. Here's a bunch of them growing in the presence of 10 millimolar manganese sulfate, and you can see that they differ in colony size. And so here are two examples of cells, just to orient you guys here. This is position on chromosome 7 from the telomere to the centromere, and this is the size of the colony when they grow in the presence of manganese. So larger colonies equals better growth. So here's one colony that went in the direction of the homozygosing in the direction of the vineyard strain. And here's one closer to the centromere that homozygosed a much larger region in the direction of the laboratory strain. So obviously from these 2 points you can't tell anything, but once you look at this for all 400 or so strains, you see a nice pattern emerge, which is that you start toward the telomere at some average value, and for that chunk of the chromosome, it doesn't matter whether you go homozygous for the for one strain or the other, right? Because that chunk of the chromosome from this location to the telomere doesn't involve any variants that influence this trait. But here, this changes where the vineyard strain, where the parent strain grows much better. Now, if you homozygose in the direction of the vineyard strain, you grow much better than you do in the direction of the laboratory strain. Pretty nice separation. And so what that tells you is that somewhere over here, there is this variant that influences this difference. And you can, this is just all looking by eye, but here are both the means of the strains separated out by genotype, and they're exactly the same here, and they separate quite widely over here. And here's a log score plot quantifying the statistical evidence for the presence of the variants of a variant at the specific location. And with very high confidence, we're recording it in this interval of 7 or so kilobases in the middle of this arm of this chromosome. So generally, though, that region still contains a number of genes and a number of variants, and generally at this point you're either done doing genetics or you have to hugely scale up your cross to increase resolution, or you have to come in with various targeted molecular techniques. But here, we can simply do this experiment again, except now instead of targeting our cuts all through the chromosome, we can target them just to this region of interest. You can see in this case we used 2 guides and we mistargeted a little bit, so about half the cuts miss the region we're actually interested in, but about half of them are exactly in the region that we targeted, giving us now very high mapping resolution. So this is the same kind of plot that I showed you before, but what's different here is the scale. Instead of this being the whole chromosome, this is a region of the chromosome that's less than 10 kilobases. And within that region, we can now refine the position of this variant of interest to a single amino acid change in this gene, PMR1, okay? So we have good recombinational evidence that separates even other variants in the same gene that are located within a couple hundred base pairs away from this location. There are 5 variants within this gene. 4 of them change in amino acid. The 5th one is synonymous, but we wanted to test it just because it's so close to the one that our mapping strategy here picked out. And we tested them directly by just making that one change, targeted change in the genome. And you can see that for changes 1, 2, 3, and 5, as predicted, they're not different from the parent strain that grows poorly. But once we flip this amino acid from this parent to this parent, you see a very substantial increase in growth in the presence of manganese. And in this case, I won't belabor this, but the gene is a manganese transporter, and the variant changes a highly conserved leucine to a phenylalanine. And if you look at the structure, the whole thing totally makes sense. I'm going to skip this in the interests of time, but we have some initial data showing that we can do the same type of thing in hybrids between closely related yeast species. So what I've described is a targeted, very high resolution method for doing genetic mapping that avoids the need for meiosis. So I'll tell you now about a second method that kind of dispenses with the whole mapping idea altogether, right? So traditional genetic mapping, as I walked you through, involves crosses, meiosis, recombination, interval. Then you have to get from the interval to the causal variant. And I just described a strategy that allows us to improve the resolution, make this variant smaller, et cetera. But you can kind of say, why are we doing this? Why do people do genetic mapping? Why can't we just like go straight to the variants of interest, right? Because ultimately, as I showed you in the last story, at the end, what you really want to do is to make a strain that contains just your best candidate variant and show that has the desired effect. That's the ultimate proof of the genetics. So why do we need to generate these mapping populations, do all of this genotyping, search the whole genome, right? Well, because you can't traditionally make that many variants at once. But we sort of started to question this assumption a couple of years ago, once again, because of the possibility of CRISPR-Cas9. So instead of doing this business, right, the idea here is, so instead of generating a mapping panel, generate a panel where the only thing that differs is you're on one genetic background, but you systematically walk through and change a specific variant from one individual's allele to the other individual's allele that you're interested in, and then just look at your phenotype, and this is the one, right? So you'd like to be able to just do that. So once again, we're using CRISPR-Cas9 to advance toward that goal, and we're not quite there yet, but I'll show you kind of our first attempt at doing this. So as many of you know, so what CRISPR-Cas9 allows you to do immediately is to make cuts, right? It generates targeted double-stranded breaks. And this can be done for a single gene, or as many of you know, it's a widely used screening tool where you have a library of guide RNAs that you throw into your pool of cells that target all the different genes in the genome. The cells all express Cas9, or it's on your vector, or however you want to set it up. And then after the cut is made, it'll be a different cut in a different gene in different cells, and the cell will repair it by just blunt end non-homologous end joining, where it'll smoosh the two ends together, and typically this will result in an indel that'll give you a frame shift. And so it's essentially the equivalent of kind of a dirty knockout screen, right? What we're interested in doing is being able to do at scale not those types of gene disruption screens, but specific allele replacement screens where we change specific variants from one to another. So Cas9, the CRISPR-Cas9 system allows you to do that by, in addition to introducing the guide RNA and the scissors here to make this cut, also introducing a repair template, right? So a sequence of DNA that has homology outside on either side of your cut, and in the middle changes the sequence to what you want, right? So this is also something that's been relatively straightforward to do with the system. But what hasn't been routine is to turn this into a screen approach, because now you have to supply, in addition to supplying the scissors, right, Cas9, you need to supply the guide targeting this, and you need to supply the repair template. And if you try to do this combinatorially by throwing in a bunch of these and a bunch of these into a pool of cells, most cells won't end up with the correct guide repair template combination, and you'll just make cuts, because you'll end up targeting the cut somewhere and then having a repair template that doesn't match it, and it'll be, really rare to get the events you want. So we managed to redesign the whole system to make something that we call mutation directing plasmids, where you have both the guide RNA and the corresponding repair template on the same piece of DNA. So then you make a library of these guys, throw them into the cells, and automatically, if you get one, you get the other one correctly. And you might ask, like, what's the big deal? Why is this hard? Why isn't everybody doing this already? So the question is, if you want to make thousands, so if you want to make a few of these guys, you just order them as a gene block or use any cloning strategy or whatever, right? That's easy to do. But if you want to make a library of, say, tens of thousands of these guys, that gets to be prohibitive. So the way these library pools typically get made is that you synthesize them in parallel on an oligonucleotide array, microarray, and then cut them off the array and then clone them into whatever your system is. And that sets a limit of about 200 nucleotides or so on the length of your construct. This whole thing is way longer than that, and the trick is to basically figure out, but a lot of it is kind of structural sequence for the guide RNA and for the repair template and so on that's not specific to any particular targeted site. And so the trick is to basically figure out how to cram everything that is site-specific into a 200 nucleotide piece and then clone that into a larger construct that has all the structural bits that are generic to any desired cut already in there. So we did that. And we showed that in yeast, using specific combinations of strains, we can get very high efficiency, in the high 90% range. I mean, the worst one of these guys is something like 95.6% of getting a replacement as a consequence of supplying these constructs. So that means that you can really do this as a pooled strategy. You make a large pool of these guys, you toss them into a pool of cells, and with high efficiency, what should come out of that is a library of cells with the replacements you want, and then you can carry out a phenotypic screen to see which of the changes matter for what you're studying. So this is what I already talked about. You get these short oligos, and then you clone them in so that the whole thing works. And there is a preprint on bioRxiv that describes all of this in excruciating detail for anybody who is interested beyond this overview. Our goal, like I said, is to take all the SNPs and other small changes from one strain of yeast and move them into another as a library. We're working on that, but we wanted to start with something simpler and where the effects were more likely to be observed on a trait. And what we decided to do is to look at premature stop codons, right? So the idea is if you generate a stop somewhere in the middle of the coding sequence of a gene of interest and you lop off a big chunk of the gene, the protein shouldn't be functional. But it is more of an open question what happens when you truncate a smaller chunk. Depending on what you cut off and how much, the protein might still work. So the interpretation of these types of mutations, even though everybody thinks of them kind of as loss of function, is not so straightforward. So what we did was we targeted premature stops to all genes that are annotated as essential for cell growth and survival in yeast. And there is about 1,000 of these essential genes. So we wanted to figure out, first of all, does this all work? Are they actually essential? And are there parts of them that are dispensable? So to that end, we targeted about 10 specific premature stops in each of these genes with a bias toward the 3' end, where you're lopping off less of it, and so are more likely to see something other than a complete disruption of gene function. And then the experiment is you have this library of cells with all of these targeted cuts. You turn on CRISPR, and then you take time points after you've turned it on and see what's going on. And if you are actually disrupting an essential gene, killing its function, the cells should be dead. So you should watch them all drop out of this pool over time. And anybody who is still there should be something that's less than a lethal mutation. So this is kind of the bulk result of this experiment. These are all the targets in essential genes. And this is basically a score quantifying how dead the cells are if they carry that mutation. So everything in here below 0 is very dead. This is a smaller control set of genes that we largely don't think should be lethal when disrupted. And you see that in general, they're not nearly as bad. Most of the stop codons that we target to essential genes are really, really disruptive, right? But not all of them. So then the question was, why not all of them? And maybe as expected, but with now some numbers on it, we see that as we plot how disruptive a stop codon is depending on where it is in a gene, and this is across all 1,000 of these genes. There is and I'm only showing you the last 150 codons of the gene because it gets really uninteresting. This line just keeps going flat. So until you get to about the last 20, 25 or so amino acids, cutting off any more than that of the protein is usually very disruptive. But out there is a basically reasonably linear trend toward a lower chance of disrupting the gene as you get closer to the end. So that's kind of across all genes, but you can look specifically gene by gene. How tolerant is that gene overall to the introduction of these PTCs? So this is the distribution of this now plotted not as a function of stop codons, but as a function of all genes. This is what it looks like for essential genes, and this is what it looks like for genes that we don't think are essential to start with. And maybe somewhat surprisingly, you see this tail of genes that look like they're quite tolerant of stop codons, even though the database says that they're absolutely essential for cell survival. So what's going on with these guys? So I've kind of gotten this annotation here, and I'll walk you through these classes. There's the conditionally essential class, the not essential class, and the truncatable class. So the not essential ones, there are 3 of them here. Basically, whatever large-scale deletion screen was done to annotate it, looks like those guys are just false positives. Like if you look at them and you look in the literature or you do the experiments, they're dispensable. There is no reason for them to be essential that we can find. So we think those are just annotation errors. The conditionally essential genes shown here basically popped up as essential in large-scale deletion screens because those strains carried mutations in certain genes required for nutrient processing, just to make the experiments easier. And it turns out that basically these genes are synthetic lethal with those genes. So for example, if you have, like the deletion connection was deletions in genes in leucine metabolism or uracil metabolism, and then a mutation in one of these other genes, those cells don't grow, right? But if you have normally functioning versions of those pathways, these mutants grow just fine, right? So these are examples of synthetic lethality as opposed to truly essential genes. And then there is a number of examples of these highly truncatable genes where, in red and green here are the calls from a hidden Markov model that we run on these guys, saying everything up to here is dead, everything past here is alive, right? And those are the individual data points of the stop codons in this gene. And this gene has a couple of different domains to it, CWC24. And it looks like you can lop off this ring finger domain with impunity. But then if you get into this zinc finger domain or beyond that, those parts really are essential. So we have a number of candidates that, that look like that, and we're just looking here at this most extreme 15 or so genes because that's kind of the highest confidence set. But we think that a decent chunk in here is probably also in this partially dispensable category. So what I told you in this second half of the talk is that we have a high efficiency CRISPR allele replacement scheme that works very well in yeast. It can be made massively parallel. And when we use it to look at stop codons, the tolerance to stop codons increases near the ends of genes, and we have some quantification of the extent to which this happens and over what distances. One thing I didn't show you is that it's generally true beyond the distance statement is if you look at what are annotated as protein domains in the databases, a PTC that doesn't disrupt the domain, so it either doesn't cut off part or all of annotated domain, will tend to be better tolerated than one that does. So if you're interrupting a domain, even if you're quite close to the end of a gene, those tend to be lethal mutations. Some genes are misannotated as essential in yeast, despite the fact that the genome was sequenced in something like 2000, and there's been a ton of work annotating it by every possible high-throughput technique ever since. And others have large dispensable C-termini, and this technique allows us to kind of do all of those annotation refinements. So I'll stop here, answer any questions, and then you guys can reach.